Larry. Some important announcements before our next keynote speaker. First of all, our exhibit hall opens 1 to 7 o'clock today, and at the exhibit hall that's the location of our expert lunches. That is the location of our poster session as well. We have a concession stand at in the exhibit hall that's open from actually 12:45 to 5 p.m. The box lunches at the experts' conversations are available at 1 p.m., the conversations with experts, 1 p.m. to 2:30 p.m. Everything after our plenary sessions this morning are in the section to your left, where the registration badges will be available for you to pick up, where the exhibit hall is, and the ballrooms for the concurrent sessions and the focus sessions for the rest of the afternoon. There'll be another break in the exhibit hall, 3:30 to 4, where we'll have refreshments. The poster viewing tonight and the exhibit hall reception will be from 5 to 7, and at that poster viewing, the presenters should be present. Also running 5 to 7 is a special reception in Ballroom H put on by the USC Keck School of Medicine. And there's a VIP speakers reception invitation only in the Gold Room on the second floor of this building from 7 to 9 p.m. There is a separate Canadian event at the Canadian Consulate for some of the invited guests, and there will be a bus to pick up those individuals in the front of the Civic Center. You should be looking at your expert lunch sheets. These will be in your conference bag and available for handout. We are adding an additional expert lunch that came on a little late. Richard Gard, the CEO of NeuralStem, will be discussing NeuralStem's ALS clinical trial at table 44, and all of those tables should be marked there. In addition, this afternoon, there are a few changes on our program that I wanted to let you know, additional speakers. On the panel at 2:30 to 3:30, there is in Ballroom a stem cell science cancer panel that is chaired by Luis Parada at UT Southwestern and Frank Pajonk of UCLA. Also joining that panel is Ellen Feigl, who's the Vice President of Research of the California Institute of Regenerative Medicine, and Dr. Stephen Chang, Vice President, Research and Development at the New York Stem Cell Foundation. Also, one of the sessions at that time, Strategies and Tactics in the Battleground States, which is item D in your program on page 5. I'm assuming personally the moderator role, and Don Reed of Americans for Cures Foundation will be a speaker. On panel F, during that time, stem cells as a powerful tool for the pharmaceutical industry. Unfortunately, Hadar Velmusky-Weinberg of Teva could not join us. Her slot will be filled by Devin Smith. Dr. Smith is a Pfizer Worldwide Research and Development. Finally, at the 4 to 5 p.m. sessions, concurrent sessions, we have a panel C, Translational and Clinical Studies of Stem Cells for Heart Regeneration, and Eduardo Marban of Cedars and Arnie Kaplan of Case Western Reserve University will be joined by Henry Sukov of the Keck School of Medicine of USC. Also, during lunch hour, we have a special workshop for a specific field. This will be carried through for 3 days, specialized workshop called IRB, SCRO, and ESCRO workshops. ESCRO means Embryonic Stem Cell Research Oversight Committees, meeting the institutional oversight challenges of stem cell research. If you are a member of any such committees, this is a very interesting program, a niche program that no other conference supplies, and that will be in Ballroom A. So I now am very pleased to bring on again one of the co-chairs of the summit, Dr. Clive Svensson of Cedars-Sinai, to introduce and a very anticipated keynote speaker, Andy Grove. Thank you.
Thanks very much, everybody. Very happy, honored, and proud to introduce the next speaker. Lucky enough to have known Andy Grove now for over 10 years. We've been involved in a few projects together, and he's an amazing guy. You're in for a big treat. Andy Grove co-founded in 1968 a small company called Intel. Some of you may have heard about. And since then, he's had various positions with Intel, including CEO, president, chairman, and he's now a senior advisor still for Intel. Intel started as a small company with a turnover of around $2,000, or the amount of money it made, revenue was $2,600 in the first year. After 30 years, it turned over $20 billion with a B, so it's one of the fastest growing companies in the world. He's known in Silicon Valley as the guy who drove the growth phase of Silicon Valley, and much loved and revered by many people within Silicon Valley. But in addition to his business skills, he's a noted author and scientist with over 40 papers, and he's authored a number of highly cited business books and highly regarded business books. And he has honorary degrees from City College of New York, the Worcester Polytechnic Institute, and Harvard University. And finally, just last week in Science, Andy had an editorial showing you how up-to-date he is on reinventing regulation, discussions about how the FDA functions, and I think you're going to hear more about that in his talk today. So a couple of quotes, and then I'll let Andy come on. The favorite one I like is, "Business success contains the seeds of its own destruction. Success breeds complacency, complacency breeds failure, only the paranoid survive." And he had a book entitled that. I very much recommend you to read it. And I think that's true in stem cells, is we have to keep innovating. Even if we think we've got somewhere, it's a very good idea to keep innovating and looking at these new techniques and new technologies. As you heard from Larry, iPS cells are a good example. We can't just sit quietly. The next one that Larry brought up again in his talk, the devil is in the details. And this comes into business. You can design things, but you have to really know what you're doing. And I think for stem cells, we have to know the cells. We have to know if they have these small genetic abnormalities. So lots of things Andy talks about will reflect in translational research. And so now I will introduce Andy. He's going to talk about his approaches to the FDA and new approaches to the FDA. Andy's never scared of saying what he thinks, and so I'll introduce Andy, and I very much look forward to what he's going to tell us. Andy Grove.
I started looking for examples of very major and complex undertakings, similar to biological projects, I'm told. And I had to go back in the past to find a good one to analyze. A few thousand years ago, if they had scientific meetings, they would be discussing the details of building the Great Pyramid, the supply chain lesson that we can learn, the number of slaves per cubic yard of pyramid, the major discovery of substituting animal fat as a friction agent as these blocks were pushed. This was before a brave soul suggested that they should use tree trunks as wheels, and I think it was thrown in front of the next pyramid block. But the most interesting thing about this, if you look at how much a pyramid like that cost, actually there's very good data. People have analyzed this in terms of man-hours, and we can take the lowest manufacturing cost of today and multiply the two, and we come with $1 billion and the time that it took to build a pyramid was about 20 years. And these are familiar numbers. Studies indicating the cost per new molecular entity, a new drug, is in the vicinity of actually $1.5 billion, and it's not quite 20 years, but high teens. More specifically, the average development time is 14 years, up from 10, and that was a few years ago, and I dare say that that number is going to be closer to 20 next time somebody makes a study like this. Even worse, and contributing to the high cost of the surviving new molecular entities, is all the drugs that we have to throw away. The attrition rate of drugs is extremely high, and it's getting to 3 nines, 99.8 to be more precise. And you all, or most of you, work on the new age of cells, genetically altered material, combination of the two. It is going to be a much harder than the basically biochemically created drugs that we based the previous statistics on. You will need new manufacturing processes. You heard some references to that. You will need, most importantly, delivery problems, because these are large molecules, and if you want to go inside the brain, which is a very important cause of societal burden of a disease, you have to figure out and have to develop techniques of consistently delivering large molecules. And that is not all. The difficulty today already is integrating the technology of the delivery techniques, the imaging techniques, the molecular analysis techniques, clinical techniques, and we can't build integrated facilities for every drug so people can do this informally. Let me give you 2 examples. One of them is the development of an implantable kidney that would take the place of dialysis and take the place of kidney transplant. The list of scientific disciplines that you have to master to do this is long. And they are esoteric, like nanotechnology, and that is before you even deal with the surgical problems of taking a contraption like this and reducing it to be small enough that it can replace the kidney of a normal person. Another examples, like I indicated to you, large molecules are particularly difficult to deliver repeatedly, reproducibly, to the exact location that you want to deliver to and nowhere else. A technique that was pioneered by the NIH scientist Edward Oldfield in 1989 to use pressure to force the fluid through the cells of the parenchyma was very promising this way, and it is still not in clinical use. And in fact, by my estimate, there are fewer people working on it today than were shortly after it was introduced. There's another indicator that I have to bring in. The pyramids cost money. There was money in the pharaoh's treasury. moving bio inventions, techniques to drugs, and qualifying those drugs costs also a lot of money, and there's no furrow. The closest to that is a VC, and they have shown a considerable disdain toward this field recently. Less than 3% of the biotech VC investment is going into biotech, and a recent Economist article explains it very simply why. It is too risky for risk capital. We are trying to work around this. We're doing strategic partnerships, alliances, reorganizations. We're trying to share the risk. Like these days, Hollywood studios don't make a movie. It takes 5 of them to make a movie because nobody wants to be responsible for it going belly up. Seems to be similar here. But all of this pushing around the risk reminds me of replacing the mud-slurry under the stones of the pyramid with animal fat. It's tinkering at the margins. Major changes are needed in this industry. And when I say industry, I include all parts of people working in academia, in government, and in the commercial industry. These changes have to be big enough that we could easily call them transformation, and just so I don't leave an undefined word, what I mean by transformation is by analogy to a phase transition, a discontinuous change of certain characteristics of the industry that renders their behavior completely different. Usually, transformations in physical phenomena and in business phenomena are caused by a large external force. And what you do at that point, when you realize that you need to do this, will not have an immediate impact, maybe even have a negative impact, but it will determine whether you go on the up curve or the downward curve. I want to talk about 2 transformational changes that I think this industry needs. One of them is to move the translational boundary closer to academia and use targeted philanthropy in a manner that you'll see in a minute. And the second one is change in its fundamental principles, the regulatory process that provides a narrow orifice at the end of this process before society gets the benefits. So the first, we need to shift more responsibility for the final product and the final outcome on the people who are the most competent in the science that we're talking about. That's, in my opinion, the medical centers. Let me walk you through in 2 slides the historical shift that has taken place in this. In the proverbial old times, things were simple. Pharma received IP from the medical centers and a royalty flowed back. Pharma put the drug on the market. Marcus Welby, MD, prescribed it, and all was well. Things got a little more complicated when pharma needed help, and this was when the transformation from classical biochemical development went to proteins, biologics, cells, genes. There were specialists forming in the world in the form of biotech companies, and to simplify things and to cut short the learning process, farmers bought them. And they usually did not work. And one of the most profound one ahead of us, which I'm very interested to see how it's going to go, is the largest biotech company being embraced and included in a large old-time pharmaceutical company. And I hope that will be an exception to that, but judging on the past, I'm dubious. I think a better method would be to take this process of moving development from pharma to the left, all the way to the medical center, form teams that involve the best scientists that you have, but also the most practical and most driven individuals, and use that integration provided by the management on the walls and the support of the institution, and aim money in the way of target philanthropy, which happens every day, but it usually happens in the form of a building. I don't remember the exact number in the opening talk, 17 new buildings as a result of CERN, if I remember right. It is easy to get donors, relatively easy to get donors to do this and put their name on a building, an auditorium, or a conference room. Why can't we learn how to motivate people to have the Joe Smith pharmacological development program? That's what we need. There's a lot of philanthropy. We need to grab it from brick and mortar and do it project. To do this effectively, we have to have the goal orientation that I implied with that organization. We have to want to operate in an integrated sense. And like I said, we have to have our funding targeted. And there is a classical example of that, where all of this happened, and I dare say a lot of us in this room benefited from it. It's the history of the Salk vaccine. We had a driven physician, a businessman turned operational manager, fully devoted to the funding part, a president who was a very effective patient advocate, and the funding mechanism that was very original, the March of Dimes. A major unspoken ingredient in the success of the Salk vaccine was that the alternatives were terrible. Acres and acres of children breathing as a result of being placed in iron lungs. The result of this program is 2 million children vaccinated in the first year of production, and polio is nearly eradicated in the United States and moving throughout the world. Let's take a look at a little bit of that.
In 1938, O'Connor didn't know much about the science of polio. What he did know was that polio patients depended on private charities like his, not the government, for help. Without a new way to raise money, his foundation could offer little more than hope. It was time, he decided, to try something bold.
The only way to fight infantile paralysis is with money. And so I'm asking you tonight to send a dime to President Roosevelt at the White House.
The March of Dimes campaign exceeded all expectations, raising $1.8 million, a staggering sum. Americans' greatest fear was polio. At his lab at the University of Pittsburgh, Salk's race to find a polio vaccine took off.
Dr. Salk's laboratory was like a factory in that he had a large number of people. They were all doing what he dictated they do.
A press release would reveal the findings everyone was waiting for.
They were going to bring it up on an elevator. The press room was overcrowded. We were elbow to elbow. The public relations man steps off the elevator with the dolly and he can't move into the press room. Everybody ganging around him. Guys were jumping over desks. The fellow climbed up on the dolly and he began flinging these things out left and right. It was bedlam.
CBS News presents a special report. The Salk polio vaccine is a success. The vaccine works.
Everybody was so happy.
A very important part of this development was the constructive way IP was used. And it's very important if we stop and think a little bit about what the patent system was intended to be and what it is. Because I think while originally it was meant as a motivator, it turned into a very strong demotivator for increased investment. The patent system was a privilege granted by the government to an inventor. The privilege was a temporary monopoly. It was intended for the inventor to go and invest in the manufacturing, being protected for a period of time by the patent right. Increasingly today, patents are used to keep products off the market. In addition to all the other problems of using simple and obvious pseudo-inventions for this, this is lethal. This is not what the founders of the patent office envisioned. It was not what existed for Thomas Edison, and it's not incidentally how Dr. Salk looked at it. While we are looking, again from The Economist, at warfare, when somebody asked Dr. Salk why he did not patent as he did not the Salk vaccine, he said it would be like patenting the sun. The next point, I think the regulatory system has to be taken apart and put back in a different form today. Some of the problems with it are fairly straightforward. It has a mission creep. It takes responsibility for more and more of the development process, the regulatory process. It uses ancient technology, and the result of the ancient technology is that the system does not scale. It isn't able to absorb increasing demand. Mission creep. In the 1930s, it was a pre-market review. Its mission was a pre-market review of drugs, and to police the famous snake oil claims. In other words, in today's terminology, it was responsible for safety. In 1962, due to a weird set of events, the law chartering the FDA was revamped, and the word requiring substantial evidence of the drug's efficacy became part of that. 1962 was the decade after the Salk vaccine, so it's pretty recent. In the late 80s and further on, it looked to me like bureaucracy run amok. 137 statutes, 18 general statutes, 14 executive orders. This is starting to sound like military procurement. Meanwhile, the trial records are largely stored on paper, in warehouses, inaccessible for analysis, for scientific secrets that are stored in those boxes, but you can get to them. And when it comes to scaling, the picture is like this. If you take the drug development and the drug regulatory system together, the numbers that characterize it in 1 year, there are 800,000 scientific papers published. That's how I get to publish one too. There are 6,000 phase 3 trials, and the infamous 20 NMEs come out. Let's try scaling the system. All your projects will succeed. You're going to have new line items, new drugs, and instead of 6,000 trials, you're going to go to 60,000 phase 3 trials. And instead of 20 patients, 200 patients. If you do a 10x, the already, I'm sorry, did I miss something? Can I go back one? Yeah. And the patients required go to 1 million. The problem with that is that recruitment of patients is already a difficult task. Somebody wrote me yesterday, he's in a clinical trial that requires a blood draw every 2 months. The trial site where he's enrolled is in Denver. He lives in the Bay Area. 6 times a year he has to go to Denver to have his blood drawn. He is in a very dangerous health condition, so he's doing it. He can afford to do this, but he is very angry. And most of the people, when they are angry, do nothing. That's why a lot of these trials die or stretch out because they can't recruit the patient. This is very expensive. A year's delay on the average costs $300 million, and it will get worse because the FDA creeps into post-marketing monitoring. Congress orders them to do comparative effectiveness research, and you all bring the additional complexity of biologics. The whole thing reminds me of the Bell Labs in the late 40s when traffic grew more rapidly than installed handsets. And the switching was done manually by people sitting at switchboards. Bell Labs knew that something dramatic has to happen because they're going to run out of people and for a switching job and level of their growth. They funded research in solid-state physics, and the result was the discovery of the transistor, which was the basis for solid-state switching systems that arrived in time to allow telephony to grow. The Bell system made a major bet on solid-state physics, which didn't exist at the time they made the bet, and they won. They survived for a long time. The child architecture that we deal with today was designed with technical requirements that were plausible in the 1960s. That has changed. How much has it changed? The cost to download a movie went from $270 a few years ago to 5 cents. Where do you think the FDA architecture is set in this picture? 2 decades or 4 decades in the past. The simplest, the regulatory environment is based on paper in boxes instead of databases. The new trial system that one might envision would use those databases to compare the individual with other people in the database very completely, with lots of items entered in at the time the drug was dispensed, compared to the entire universe in the database faster than you can even think. And because of the architecture, not only will that person know his or her sensitivity to a particular drug or a side effect as rapidly and as easily as you can find your reading preferences, movie preferences on Amazon.com, but you can make all of that data open for research by looking at it as being the property of the community that funded our existence in science. The best thing that I can hope, let me go back. There are signs of this type of thinking appearing. There was a very interesting editorial recently in the Wall Street Journal, which actually is a leading publication in this kind of thing, calling it a new bargain for drug approvals, arguing that returning the FDA to its earlier mission of safety and leaving proof of efficacy for post-approval is the direction they should go. I never thought I'm going to argue my case with a slide taken from a libertarian think tank. It's called, borrowing Milton Friedman's concept of free to choose as the basis for our society. They call it free to choose medicine. They argue leave the existing drug flow present, but give them a shunt where you can, if you wish, you can go around and give up confidence in the drug for certainty of its availability while it can do you some good. You had some discussion in this room where you heard the voice of a patient. My interpretation is, I don't know who that person was, but I think it was a female person. She would choose the shunt. We also heard a considerable discussion of the shunt being trips to unproven foreign labs. Are we maybe responsible forcing people into less desirable shuns by not providing one ourselves. Today, the Wall Street Journal, can you put the camera on? I don't suppose you can read that. An opinion piece by the Deputy Commissioner of the FDA from 2005 to 2007, and a practicing physician and investor. Title, How the FDA Could Cost You Your Life. You can imagine the general content. So the question is, where do we go from here? How do we stimulate an action that requires governmental action, financial changes, organization structures of medical research? I don't have an answer for that, but I have lived through corporate and industry transformations in the field that I got my training in. And what I learned is these transformations are more difficult when there is no emergency. Polio was an example in the other direction. When we don't know what to do, we should experiment. Sitting at a desk will not tell us what to do. I have a way of describing it. We should let chaos reign for a while, and when we develop more understanding, we should reign in chaos. This is the next one is the one I don't know what to do about. In a corporation, there is central leadership. Sometimes they don't exercise their right and obligation to lead, but other times they do. Industries don't have CEOs, and yet we require an industry-wide momentum to do this. Experimentation is easy. The reining in on an industry level, I don't know how to do. The only central force is the state, and all of us have been trained to object to the state monkeying around in our business, and we were trained to yell industrial policy and run. Most importantly, I don't know who will drive this change. Politicians, insurance companies, very possibly because they are smelling change from a distance and move, or the medical professions. I would like to think of the last one. My question is, are they and are you, as being a building block in a very important avant-garde part of the medical profession, are you up to it? Stem cell research was born in politics. You are probably more able to visualize the government or another force guiding you or stopping you. And if you know how to multiply, you come to a conclusion that in a few years you will be absolutely dammed up in that orifice that the regulatory process represents. So the question can I go back? The question that I would like to leave you with: are you ready to be an accidental pioneer? Because you did not bargain for that, but your career depends on it. In the next slide, as you started to read ahead of my intention, you can read now. These are the alternatives that you can follow.